Oh, come on. Okay, before we begin, are there any questions? Not hearing anything, so let's begin. Let me share my screen. It's not working normally. There we go. Guess I can close that down. I might want to show you this. All right, on the syllabus, we see that today we're going to be working on Lab 9, identification of gram-negative organisms. Let me remind you, you have a quiz this Wednesday in the lecture, which covers both the lecture and the lab. On the rest of the labs, you're only going to be working on your unknown project. I will be logging in for the first 15 minutes of the lab to answer any questions. So if you have questions, especially about the lab, but any questions, I will be logging in for the lab for at least the first 15 minutes. But if there's no questions and nobody logs in, after 15 minutes, I'll log out. Let me remind you, your unknown project is due Saturday, March 9th at 11:59 p.m. Your unknown notes pages will close on Friday, March 8th. If you request a test, and the last day to do this is on Friday, March 8th, if you request a test, it must be a test that will incubate for a day or less. Otherwise, you will not get the results in time. Any question about any of that? All right. No, this isn't here. We're going to have the review, if all goes well, in the lecture on Thursday, the last week of the term, before the final. It's on the Monday, Wednesday class. I have a review on the lab schedule, but that was because they lost a day. All right, any questions about anything? If not, I'm going to close that down because my computer isn't working well. Not sure what the problem is. Let's go to the lab. Blow that up a little. So in today's lab, the identification of gram-negative bacilli. There is some reading to do, just a page on biochemical tests and bacterial identification. 3 or 4 pages on methods of classifying and identifying microorganisms. You should read that material before you do the lab. There are some video clips in today's lab. The learning objectives are, upon completion of this lab, you should be able to, 1, interpret the results for the following biochemical tests. The IMVIC reactions. IMVIC is an acronym, I for the indole test, M for the methyl red test, V for the Voges Proskura test, and then C for the citrate test. These tests are used for identifying, especially the Enterobacteriaceae, but generally the gram-negative bacilli. We're also going to talk about the carbohydrate fermentation tests, or test, depending on how you look at that. We'll say tests because there's several of them. The motility test and the hydrogen sulfide production test. Any question about any of that? 2, you need to be able to explain how to interpret the TSI test, that's the triple sugar iron slant, the SEM deep test, or the SEM test, and that's the sulfur indole motility test, And then the test of growing cells on McConkey's agar, MAC, or hectone enteric agar, HE agar plates. All of these tests are running multiple tests at one time. For the SIMS test, we're running the sulfur, the indole, and the motility test. That one's pretty easy. McConkey's, we're looking at whether the bacteria can grow on the McConkey's plate. And if it does grow, it's likely to be gram-negative. And then you can determine the sugar fermentation of lactose on McConkey's. And the same thing for hectone enteric agar, only gram-negative can grow on HE plates. And we're assaying for the fermentation of sugar. HE plates also can assay for hydrogen sulfide production. And then 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. Any question about any of that? Oh, come on. Told you my computer isn't working too well. We're going to be talking about 2 main families of gram-negative bacteria in this lab, the family Enterobacteriaceae and the family Pseudomonaceae. Both of these families are small gram-negative bacilli. The family Enterobacteriaceae are non-spore forming. They are all facultative anaerobes. They all ferment the sugar glucose, and they all are oxidase negative. Any question about any of that? This family is one of the largest, and there are 53 genera in the family. 24 of these genera are known to cause human infections. These genera shown here are usually normal microbiota, meaning they grow in the intestinal tracts of animals and humans, at least some of them, and therefore we call them coliforms. Like Escherichia, and I think Citrobacter grow, and Enterobacter grow in our intestines. Salmonella and Shigella do not normally. Salmonella, though, does grow in the intestines of birds, maybe lizards too, I'm not sure about them, but definitely of birds. And that's why when you're handling chicken or any other bird, you should be very careful because salmonella is likely to be on the bird. And if humans were to get salmonella, it would cause disease, gastroenteritis. So all of these members of these genera are opportunistic pathogens, meaning they grow, at least in animals, in the intestinal tracts. Many of them grow in the intestinal tract of human, but they can cause urinary tract infections, wound infections, pneumonia, and septicemia. So if they grow where they shouldn't be growing, it can cause a opportunistic infection. Common genera are responsible for causing opportunistic infections, including Escherichia, Enterobacter, Klebsiella, Citrobacter, and Serratia. So these are often in humans' intestines, like E. coli and Enterobacter, Citrobacter, maybe Serratia, I don't remember. But if they grow in other cases, it's an opportunistic infection. Grow in other locations other than the intestines. And they have frequently caused, that's not right. There are other members of the family, that several genera that cause gastroenteritis. And if humans were to get them, like Salmonella, Shigella, or E. coli, Serovar 015787, they will cause gastroenteritis. These to humans are frank human pathogens. So you do not want to get them in your gut or you'll get gastroenteritis. And E. coli serovar 0157H7 has the antigens 0157H7. These antigens are human pathogens. Is that what I'm trying to say? E. coli that has these antigens is pathogenic, causing gastroenteritis. Normal E. coli does not have these antigens. And normal E. coli does not cause gastroenteritis. In fact, we have it in our guts and it's important to help us digest our food. But there are certain variants of E. coli like serovar 0157H7, which is a frank human pathogen. Any questions about any of that? All right, that's also it for the discussion of the family Enterobacteriaceae. Let's talk about a little bit about the family Pseudomonaceae. This family is similar to the family Enterobacteriaceae in that they have the same Gram reaction and they're the same shape, meaning they're all small bacilli, Gram-negative bacilli. However, the Pseudomonaceae family are all non-glucose fermenting. The Enterobacteriaceae all ferment the sugar glucose. And then the Pseudomonas are all oxidase positive, and the Enterobacteriaceae are oxidase negative. Pseudomonas aeruginosa is a human pathogen, but it is really lousy at getting established in an individual with normal intact skin, mucous membranes, and a normal immune system. So it's not very likely to get established on a, quote, normal individual. However, an immunocompromised individual, a burn patient, or a trauma patient can frequently get an infection from Pseudomonas aeruginosa. The burn and the trauma patients will frequently have damage to their mucous membranes or their intact skin, allowing Pseudomonas aeruginosa to get established. And then immunosuppressed individuals are more likely to get any bacterial infection, but particularly Pseudomonas aeruginosa tends to hit them. And it's probably just that because Pseudomonas aeruginosa really is almost everywhere. And we are constantly exposed to it. It's just very lousy at getting established and causing an infection in, quote, a normal individual. But it is often a healthcare acquired infection. We talked about those previously as a nosocomial infection. It is not the number one. E. coli is actually the number one nosocomial infection. But Pseudomonas aeruginosa is a frequent nosocomial infection. You can find Pseudomonas aeruginosa almost in anywhere where there is moisture, such as a wet bar of soap, perhaps dilute detergent or soaps. Like if you had your soap in the dispenser at home for over a year, it may have Pseudomonas aeruginosa growing in it. I've been tempted always to test the detergent in the microbiology lab to see if Pseudomonas aeruginosa is growing in the detergent because it is dilute and we always have it in the microbiology lab. And so it's always in the container. And when the container is empty, we just refill it. So it's a chance that it's in the detergent, even in the microbiology lab. And then in hospitals, it's in things like flower vases, in dirty mop water, in a sink that is got moisture in it. Any questions about Pseudomonas aeruginosa? All right. Let's move on. It's important for microbiologists to be able to identify and perform tests on gram-negative bacilli, especially if it's submitted for a lab for as a clinical specimen to be tested in the lab. In this lab, we're going to talk about several of the biochemical tests that are used to identify gram-negative bacilli. Here, we're looking at a whole bunch of tests which can be used to identify gram-negative bacilli. We're not going to talk about all of these. And this is something I just want to briefly talk about. This is a bunch of tests. And what you do is you get the pen, which is, it looks like this one is pulled out of here. And you inoculate the pen, it's sterile, get the bacteria on the tip of the pen. Hopefully you can see this. And then stick the pen in this device, sticking the pen all the way through 12 tests, and then you can run 12 tests at one time. And those are the different tests up there. We're not going to talk about all of these tests. We'll only talk about a few of them. The first test we're going to talk about is the indole test. Some organisms can produce the enzyme tryptophanase that can convert the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added to the tube. Kovac reagent reacts with indole to produce a red color at the surface. E. coli tests positive for indole production. Klebsiella does not. It tests negative for indole production. Oops, let me scroll down. So tryptophan in the media, and that can be either media that has that amino acid added to it, or it could simply be a medium, I guess I should say, with a lot of protein in it. And the protein can have the amino acid tryptophan. And so the bacteria can digest the protein and then convert the amino acid tryptophan to indole. When you add Kovacs reagent, it should be a cherry red color. There's a little bit something wrong with this this picture, and it should be cherry red in color. That looks more like a purplish color. Usually it's cherry red. All right, any questions about the indole test? If not, let's move on to the MR and the Voges Perskura test. I'm just going to call it the VP test for short. The MR test or methyl red test and the VP test are usually in the same tube. And then you inoculate that one tube with your test organism, grow up the media, and then you split the tube into two parts. One part being used for the MR test, the other part for the VP test. So usually you inoculate that together in one tube. Any question about any of that? So both tests obviously have the same media in it. They're both looking at the results of glycolysis. The MR test is looking at acid end products of glycolysis. The VP test is looking at neutral end products of glycolysis. All right, the MR test requires 5 days to incubate. You could grow it for 2 days, but if the test is negative, then you have to repeat the test and grow it for 5 days. So generally, when people ask for the unknown results on the MR test, I just let it incubate for 5 days. The VP test can be done in 2 days, but there's no harm in doing it for 5 days. And usually what we do is we just inoculate one tube, let it incubate for 5 days, and then split the tube in two, one part being used for the MR test, the other part for the VP test. Methyl red is added to the MR test. This is a pH indicator, and if acid products are made, the methyl red will remain red in color. Acid products happen by the metabolism of glucose. Fermentation of glucose makes acid products. If the methyl red changes color from yellow, and that usually happens immediately. You put in the drop of red, methyl red, and it's in the tube, and it immediately changes color to yellow. If it does change color to yellow, it is MR negative. So let me show you the results down here. So this one, we added the methyl red test and it remains red, so that's MR positive. We added the methyl red here, the color changed to yellow, and that's methyl red negative. Any questions about methyl red? All right, the VP test, you add Barrett's reagent A to the tube for the VP, meaning the half of the tube that is for VP. You shake the tube, then add Barrett's reagent B to the tube, shake the tube again. The purpose of shaking the tube is to add oxygen. So you need both Barrett's A, Barrett's B, and oxygen in the tube to get the reaction occurring. You let this run for 30 to 60 minutes. And if acetone is made from glycolysis, that's the neutral product, it will react with the Barrett's A and the Barrett's B and the oxygen to produce a red color. If the acetone is not made, the color will remain yellow and will not turn red. And so that would be VP negative. This would be VP positive. Any questions about the VP test? All right, the nice thing about all three of these tests, the MR test, the MR test, the VP test, and the indole test all turn red if they are positive. And like I said, that's normally a cherry red color, not a purple color. And then if they're negative, they're all yellow. So that makes these three tests a little easier. Any question about any of that? If not, let's talk about the citrate utilization test. This test is used to determine whether the bacteria has the ability to metabolize citrate. The only carbon source in the citrate utilization test is citrate, that sugar. There is no other carbon source. So if the bacteria cannot utilize citrate, it cannot grow on the tube, and the color of the tube will remain green. So that's citrate negative. If, on the other hand, the bacteria can metabolize citrate, citrate is a sugar in the Krebs cycle, we will get citrate metabolized, the bacteria will grow, and the metabolism of the sugar citrate will produce a basic end product, which will make the tube alkaline or basic, change in color from pH green, meaning this is normal around pH 7, and then this is basic pH above pH 7. I'm not sure what the pH is, it doesn't matter, but so if the bacteria can grow on citrate, you should see growth. You can't see growth here. And then the tube will turn blue. Any question about any of that? E. coli and Shigella cannot metabolize citrate. I believe it's Citrobacter furundi can metabolize citrate, will grow on it, and will be blue. All right, let's talk about the PR carbohydrate metabolism test. This test is looking for the fermentation of one sugar, which is in the media. So we could call this the PR glucose test, or the PR sucrose test, or the PR mannitol test, rather than calling it all those different names. I will simply call it the PR carbohydrate test, because one sugar is in there, and that could be glucose, fructose, sucrose, lactose, mannitol, inulin. We could test all of those for the unknown project, except for fructose. That's not in the table, but we could. And you look to see if the sugar is fermented. If the sugar is fermented, fermentation will produce acid end products, and that will change the pH, making it acidic, changing the color from reddish or pink to yellow. And so this one we would abbreviate A for acid, and this one negative, or maybe K. K stands for alkaline, negative meaning there's no reaction to the tube. If fermentation occurs, some species can ferment not producing a bubble of gas, and then other species can ferment and they do produce gas. If you have gas production in the carbohydrate fermentation tube, you'll see a bubble in this inverted Durham tube. And the inverted Durham tube should have media inside the tube. So this you would abbreviate AG for acid and gas. Any question about the PR carbohydrate metabolism test? If there's no questions, I'm going to talk about the motility medium. The motility medium is a media containing agar, but the agar is at low concentration, so low that the agar sort of jiggles like jello. If the bacteria is not motile, meaning it does not have a flagella, you stab, inoculate the media, and then pull the needle directly out the way you brought it in, so it's only got one clear stab mark. If the bacteria can only grow in the stab, then that bacteria is non-motile. And that's shown here, non-motile bacteria. If, on the other hand, the bacteria can flare out from the stab, I'll draw the flare here. It may be different on this side. So if the bacteria has a shape where there's a flaring out of the media, that means the bacteria is slightly motile. The bacteria hasn't gone everywhere. It's still near the stab mark, but it's definitely flaring out from the stab mark. So it is slightly motile. If the bacteria is really motile, you'll see the bacteria go everywhere in the tube. You won't be able to see the stab mark, but it's right there. The bacteria is everywhere, and so the tube will be cloudy everywhere. And there's a clear tube where you can see the media has no bacteria in it. Obviously, it has it in the stab mark. And this one is cloudy because the bacteria has swam everywhere and is making the tube look cloudy. So that would be very motile bacteria. Any question about any of that? For assaying for motility, we usually just say negative and positive. So if it's slightly modal or very modal, we just record it as positive. But you could break it down into very modal and slightly modal. Remember, if bacteria are modal, they have to have the flagellum or flagella, because that's the only cell structure bacteria have for motility. That isn't true for eukaryotes, but we're not talking about eukaryotes here. Any question about the motility test? If not, let's move on to the hydrogen sulfide production test. If the bacteria can produce hydrogen sulfide, there are several tests to detect the hydrogen sulfide being produced. We could test it with the triple sugar iron test, and that could also be called the TSI test. We could test it with the sulfide indole motility test, or SIMS test. We could also test it on the hectone enteric agar. If the bacteria grows and produces a black precipitate, then hydrogen sulfide gas has been produced. What happens is the reagents react with the hydrogen sulfide gas, forming a black precipitate. Citrobacter, Salmonella, and Proteus produce hydrogen sulfide gas. E. coli normally does not produce hydrogen sulfide gas. Here we're looking at the SIMS test, and it was stab inoculated. And as you can see, this tube is positive for hydrogen sulfide production, while the tube changes color to black. You could also run the hydrogen sulfide test on the triple sugar iron test shown here. Let me show you some results of that. So here is black. That's black because of the production of hydrogen sulfide gas. And this is the TSI, or triple sugar iron test. You could also test for hydrogen sulfide production by growing it on HE plates. And if the colonies turn black, then the colony cells are producing hydrogen sulfide gas, which allows the formation of the black precipitate. If, on the other hand, the colonies are their normal color, like here, they're normally white or off-white, and then they grow on the HE plate, they're likely to be a gram-negative cell that does not ferment the sugar, does not produce hydrogen sulfide gas. So no hydrogen sulfide gas production here, hydrogen sulfide gas being produced here. Any questions about any of that? All right, if there's no questions about that, let's talk about the triple sugar iron test, also called the TSI test. This test shown here has 0.1% glucose and 1% lactose and 1% sucrose. So we'll be looking at the fermentation of these sugars in this test. The test also will be looking at the production of gas. If you see gas, you'll get a bubble or cracking of the media. I'll show that here. Obviously, there's a big bubble. There's a little bubble right there. And then there's cracking of the media. There's a little bubble right there, too, on this side. So this cracking of the media is due to the gas being formed. Usually, you don't get so much gas as this one, but sometimes you do. The test is also looking at the production of hydrogen sulfide gas. If it is produced, the tube will turn black as shown here. We are also looking at the growth in this tube where it'll grow aerobically, and that growth will be on the slant here, aerobic growth, and we're looking at anaerobic growth. It may only be partial anaerobic growth, but that would be the growth of the bacteria inside the butt of the tube. So those are all the tests we're running in the triple sugar iron test. Any question about that? How you inoculate the triple sugar iron is you get a needle, which is actually shown. You might have to look at lab 2 to get the needle. I don't think I have it here. But anyways, it's like a needle and thread, meaning a straight device. Actually, there's the needle there. I knew it was shown someplace. I just couldn't find it. What you do is you get the tip of the needle in the bacteria, and you first stab the butt of the tube with the needle, and you should go down 2/3 of the way. This looks like halfway from the bottom of the slant to the bottom of this tube. You don't want to go all the way down, because if you touch the bottom of the tube, you'll damage the needle. And then you pull the needle straight out the way you stuck it in, so that it has a real nice defined hole where you inoculated the tube. And then that will test for growth anaerobically, or at least partially anaerobically, in the butt. You then pull the needle out and then pick up bacteria once again on the tip of the needle, and then you streak the slant of the TSI tube to grow bacteria on the slant, and this will be where we're looking at the growth aerobically. So this will be aerobic growth, and this will be anaerobic growth. Any question about any of that? All right, the results of this tube are complex because there's many things going on. In tube 1, we get no change over no change, and you put the reaction on the slant over the reaction over the butt. And so in this case, they're doing it K over K. K stands for alkaline in the slant, K alkaline in the butt. You could write that negative over negative, meaning no change in the slant, no change in the butt. Any question about tube number 1? If not, in tube number 2, we have pink or no change in the slant, so that's going to be K or negative. And then in the butt, it's black, so we'll put hydrogen sulfide being produced. We can't see if the butt is yellow or not, but I'm going to tell you that if hydrogen sulfide is being produced, the bacteria are fermenting the sugar in the tube. So if we had looked at this earlier and the black wasn't everywhere yet, then the edge around the black would have been yellow. So whenever it's black, you can assume, in TSI, you can assume that it is yellow in addition to being black. You just can't see the yellow, at least at this point in time. There's no gas being produced. There's no lifting of the auger, no cracking of the auger, no hole in the auger. With tube 2, only glucose is fermented, and I'll talk about that when I talk about tube 3. Let's move on to tube 4 because it's the next easiest to explain. In tube 4, we have on the slant, a yellow color, and in the butt, a yellow color. So we abbreviate that A /A for A, acid being produced from fermentation in the slant, and then A for acid being produced in the fermentation of the sugar in the butt. In tube 4, we know that both glucose is fermented and at least one other sugar. It could be lactose, it could be sucrose, or it could be both of them. Exactly which sugar is fermented, we do not know, because there's just too many things going on in this test, and it's hard to answer this question, which sugar is fermented when the tube is yellow. The point is you abbreviate that A over A, and the bacteria is fermenting the sugar in the slant as well as the butt. Any question about any of that? If not, let's talk about the more difficult one, and that is tube 3 is pink on the slant. So we write K or negative for the slant, and then yellow in the butt, because the butt is yellow and the slant is red or pinkish. We know that one sugar is being fermented. That's why the butt is yellow. But probably if we had looked at this earlier, we would have seen that the slant was yellow. And that's because this media does have peptone in it. And if the bacteria can only metabolize glucose, glucose is held in this tube at low numbers. And if glucose is used up by some other microorganism, and then you're testing a second organism in the TSI test, if you were to get this result, it is negative or K alkaline in the slant, because the only glucose was fermented, and the glucose is fermented quicker and it grows quicker on the slant than it does anaerobically or partially anaerobically. And so that's why the butt is yellow, but the slant is not. And the slant is pink because the bacteria, rather than dying or just going into stasis, not doing anything, it will metabolize the proteins for its food source. The protein being in the media, along with the sugar. And if the bacteria ferments or metabolizes the sugar lactose, then, or any other sugar for that matter, if it is red in the slant, the sugar has been used up, and so it cannot ferment any other sugar. And that's why we know this is glucose, because glucose is at lower concentration. We do have sucrose and lactose in the slant, but it's not fermenting them, and they are at 10 times the concentration of glucose. And the cells will grow quicker here, use up all of the glucose, and initially it will turn yellow, but during the time span of this test, the bacteria will start metabolizing protein and then producing what is it? Losing what that is called. It will produce ammonia. There we go. It'll produce ammonia. For some reason, I lost the name of ammonia. And ammonia is a base. And even though that was yellow to begin with from the fermentation of glucose, now the protein will be digested, producing ammonia. Ammonia will be excreted by the cell, will go out into the slant and change the color to red or pink, because ammonia is a base. However, the bacteria grow slower in the butt, and this is because it's anaerobically, and they also metabolize the glucose slower. So there's still glucose in the butt. It hasn't all been metabolized. That's why it's yellow. And while the glucose is being fermented, it'll produce an acid product, why it's yellow. And that's why we have a tube where it is reddish or pinkish on the slant and yellow in the butt. It's because only the sugar glucose was fermented. Any question about the TSI tube? The TSI test. All right. And that's also the case for the slant here. Only the sugar glucose is fermented. It's black down here, but we can assume because it's black that it has to be yellow as well. And that, I don't know if I mentioned it, is because hydrogen sulfide is produced only when the cells are engaging in fermentation. All right, let's talk about the next test, the sulfide indole motility test, also called the SIMS test. What you do with these tubes is you have a media and you inoculate the media. And there you can see the stab. It's probably best right here. There it is, right there. You can see the stab. If the bacteria cannot move outside of the stab, then we know the bacteria is non-motile. It lacks the flagella to allow it to swim through this media. And this media is very thick, but it is not solid. I don't remember if it's semi-solid or not, but it's not solid. If it is motile, you see it flaring out from the stab. If it's non-motile, the bacteria will only be in the stab. And I think that one's non-motile, but I'm having a hard time viewing it. If it's black, that means that the bacteria has gone everywhere in the tube. You can't see that because of all the black color. But if we were to look at this earlier, maybe the black would only be around the stab mark, and then the edges would have been showing bacteria moving out from the stab. But that is motile. Any questions about the motility or the hydrogen sulfide production in the SIMS test? There's hydrogen sulfide positive, all the others hydrogen sulfide production negative. In this tube, we also add Kovacs reagent to the top of the tube, and that's usually not a problem because the reagent is lipid soluble, so it will float over anything that's mostly water, like the media in these SIMS tube. The Kovacs reagent, if it turns pink, it means that indole was present and produced by the sugar. So this tube is indole positive, so is this one, and so is that one, and so is that one. But this tube and this tube are indole negative, and they will be yellow in color, pale yellow and a brighter yellow. Any questions about SIMS tube or the SIMS test? If not, let's look at the McConkey's and Hectone and Terret agar. I'll just call that MAC and HE plates. If bacteria is growing on these two plates, then that bacteria is likely a gram-negative. And that's because both MAC and HE select against gram-positives, but they let gram-negatives grow. If you do get growth on either plate, you can then assay the plate for the differential components. And that is, we're looking at the metabolism of the sugar lactose. If the McConkey's plate cannot ferment the sugar lactose, NLF means non-lactose fermenter, and that's in the key here. There we go. So the bacteria is a gram-negative, but the one on the right is not fermenting the sugar lactose. The one on the left is gram-negative, and it is fermenting the sugar lactose, and it will have pink or pinkish colonies, as well as pink around the media. Any question about that? So lactose fermenter, gram-negative, non-lactose fermenter, gram-negative, and I guess I said it, non-lactose fermenter. Let's move on to the HE plates. HE is also selective. So if there's growth, and there is, then we know it's a gram-negative. If it turns black, and we're talking about the colonies, then we know that sulfur is being metabolized. If it is not black, and the colonies are its normal color, here it would be either white or off-white. That means this bacteria is likely to be a gram-negative, and it's not fermenting the sugar lactose. NLF means not fermenting the sugar lactose, or non-lactose fermenter. If the bacteria ferments the sugar lactose, the bacteria will have a salmon color to them. Salmon is sort of a yellow-orange color. And this bacteria growing on HE is lactose fermentation positive. If ever you have both black and lactose fermentation going on in the plate, you cannot tell by the color of the colonies, because they will be black from the presence of the lactose fermentation on McConkey's, or not McConkey's, HE. However, what you can do is, even though the colony is black, you'll note that the media around the colony is an orangish or a yellow-orangish color. And so if you see this color around the bacteria, you know that this bacteria is a lactose fermenter. Because if it was not a lactose fermenter, it would just grow in the colonies, and there would be no orange or purplish color around the area where you have either your shield or the area that you've sampled with one of your captains. Any question about any of that? If not, let me shrink that down. All right, so let's talk about Laboratory 9. You're not going to be physically performing any of these tests because our class is online, but you are responsible for knowing how to perform and interpret these tests. We have some video clips for you to watch. On the first, there's carbohydrate fermentation. On the second, there's the SEM test. And remember, that's engaging in 3 different tests at one time: hydrogen sulfide production, indole production, and motility. We'll also look at the MRVP test. Remember, this test is inoculated as 1 tube, and then we split the tube into 2, running the MR test on one half of the tube and the VP test on the other half of the tube. Anyways, we have a link for watching the MRVP test. We have a link for watching the TSI test. And remember, that one has a lot of things going on. I'm not even going to try and remember all of them, but there's fermentation of glucose as opposed to the fermentation of glucose, lactose, and or sucrose. You can look at the growth anaerobically and look at the growth aerobically. You can use the TSI test to test for the production of hydrogen sulfide gas. You can use it to test whether the gram stain is gram negative or gram positive. And then lastly, you can use the TSI test to look for the production of gas forming a bubble or cracking of the media in the tube. Any question about any of that? All right, we have a video on McConkey's auger plate, so watch that video. When you're all done with that, then click on this link. Sorry it's so small, microbuzz. Open the page. Trying to get there. Not there. I don't think I have it up yet. I just clicked on it too. Like I said, my computer is having trouble. I'm not sure what the problem is. It's not letting me open it. Click on the link for microbuzz. Let me blow that up so I can see it better. And then you need to click on the link for the differential tests over here. Only the differential tests are we going to talk about. And for the tests of the following table, if you need to look up the results, you can get them from mostly the microbuzz website. Obviously, some of the tests you can get from this lab module. Let me go to that now. Oh, there it is. So in question 1, you need to fill in the results for each member of the table. The very first cell has been filled in for you. For the oxidase test, if the test is positive, the cell or the spot will turn blue. And then provide the negative result over here, and then move down to indole. Indole, methyl red, VP, citrate, carbohydrate fermentation, sugar fermented, gas produced. All of these you can get from this lab. A TSI auger slant, triple sugar iron test, looking at glucose fermentation or lactose and sucrose fermentation. If only the butt is red and only the butt is yellow and the slant is red, you know that is positive for the sugar being fermented. And hopefully that'll help you identify. Anyways, each cell in the table, make sure you put in the answer. For all of these, you can get it from the lab manual. But for these down here, oh, there's one. Oxidase. I don't think we talked about that today. But it seems to me we did talk about indole, methyl red, VP, citrate, carbohydrate fermentation, the TSI, the SEMS, and the MAC and the HE. So to get the negative result, you could go look at microbuzz if you don't know. And I'll tell you the negative result is the cell or spot does not turn blue. It stays its normal color. Try and be brief in your answer, but if you need to write in a phrase, go ahead. Do not give me an entire sentence. Try to truncate it into a phrase. And most of the time, you can just put plus or the color. I guess this is the color for the positive result for VP. So you'd say blue in color, for example, and then over here, the negative result. So fill in this table for question number 1. Then go down to question number 2. And we have a whole bunch of questions here on 2A. What reagent must be added to detect the presence of indole? So write the reagent there. 2B, methyl red is a pH indicator that remains what color under acidic conditions. So give me the methyl red color under acid conditions. 2C, what test uses Barrett A and Barrett's B reagents to determine the presence of acetone, a neutral product formed from glucose metabolism? So give me the name of the test there. For 2D1, Simmons citrate uses a pH indicator that turns blue. So name the pH indicator right here. And then it turns blue under acidic or basic conditions. So answer that it's blue under acid or basic conditions. You may simply bold the answer. You may underscore the answer. You may also write the answer like over here. Any question about any of that? All right. For 2E, what is the pH indicator used to detect PR carbohydrate fermentation? And is yellow under either alkaline or alkaline or acidic conditions? So for 2E1, tell me the name of the pH indicator, and 2E2, tell me when this test is yellow under acidic or basic conditions. Any question about any of that? All right, so make sure you answer both. Once again, you can bold your answer or underscore your answer, or you can write it just right there. Oh, come on. All right, for 2F, bacteria that possess this structure test positive for motility. Name the cell structure there. And I think I changed that just today. I changed it to bacteria that possess this structure. Just before I just said like an organism or a cell. And eukaryotes have other structures for motility that bacteria lack. So just give me the structure there. Make sure it's a cell structure. 2G, 3 organisms that produce hydrogen sulfide are, give me the names of 3 organisms that produce hydrogen sulfide. They were mentioned in the lab module. Another place to get them would be look up on your table of unknowns. 2H, an organism ferments glucose but not lactose or sucrose. So the TSI slant will be what color, and the deep will be what color. Any question about 2I? In 2J, we have MAC and HE plates, and they are both what in that they inhibit the growth of either gram-positive or gram-negative bacteria. So tell me that they're both what? Tell me what that word is. And you do need to write that in. And then you can bold or underscore or write in that MAC and HE plates are selective against gram-positives or gram-negative. If you want to write it, probably write it at the end of the question. And then you can just bold or underscore it. These plates are also what in that they can separate various bacteria based on visual differences from sugar fermentation. Any question about 2J? Remember 2J has 3 answers, 1, 2, 3. And I suppose that's the same as 3I, but that's a little clearer that it has 3 answers. All right, once you're done with that, then complete the case studies and use this key to help you identify which species is causing the case study. Only use this key for this lab. It will not work on your a known project. For one reason, it's only got 2 families here and not that many species in both families. Yep. Then scroll down to this table here. This table is for you to put your results from case study 1. Like if they tell you that it is oxidase, you can put in oxidase positive. And for there, you can't abbreviate. Like if you want to say oxidase positive, you could just put plus. But if it's oxidase negative, you put not minus. And you can use yes and no. Any question about that? So from your case study, if they discuss glucose, put your results there. If they discuss lactose fermentation, put your results there. If they discuss the TSI, put your results there. And this one will be a little hard. You may need to blow that up if you have a lot of text, but you can abbreviate it. What it's really looking for here is the something like the acid / acid, meaning put your slant results over your but results. You don't need to talk about hydrogen sulfide here because we have it in the next column, and then motility in the next column, indole production in the next column, MR and VP, the next 2 columns, and then we end the test with the citrate test. Any question about any of that? Once you get your results in the table here, you can then look up on the table and figure out which gram-negative bacilli the species is. And it should be a species. There might be one exception. I think there is one exception, and that is a case where you have to write something in there. But other than that, you should be able to just abbreviate sort of like this on this table here. You will not have every test on every case study, but you will have many of them on each case study. And then use this key to identify the species causing the problems in the case studies. Let's briefly look at the case studies. They're in the back part of the lab module. Oh, I got the wrong one again. Let me look for the other one. All right, case study 1. You can read about the individual, and then they grew up the bacteria from him, and that's on the bacteria on a blood agar plate. You can see the hemolytic pattern, and then they streaked out the bacteria on McConkey's plate, and the fact that it's growing should tell you something. The fact that it's pink should tell you something. They then did the gram stain over here, and if you need to, you can blow that up so you can quickly see the bacteria. Come on. Let me blow that up even bigger. Come on. They're very small, which is why I had to blow it up so big. It's a little blurry, but you can see right there the shape of the cells and whether they're gram negative or gram positive. This is a gram stain, by the way. All right, let me shrink that down again. All of the bacteria in this lab should be gram negative, and it should be a gram negative member of Escherichia family or a gram negative member of Pseudomonas family. All right, Let me talk briefly about case study. Well, wait a minute. Let's finish case study 1. And that's the first results, growing up the cells and doing the gram stain. We then have some other results here. And sorry, this page is broken. The key for the results. So this is the SIMS test, and there's the result there. This is the MR test, and there's the result there. This is the VP test. There's the results there. There's the Simmons citrate test. There's the results there. And this is the triple sugar iron test. And there's the results there. You'll notice that this is yellow here, yellow there in the butt. It's not black anywhere. And there's cracking of the media. And right there, we have a small air pocket in between the bottom of the media and I guess the top of the tube, meaning not the top, but the inside of the bottom of the tube. Any questions about that? Let me read just a little bit about case study 2. Mikey talks his mother into buying a pet turtle. He does well at changing the water, but he's not diligent about washing his hands after touching the turtle. About 2 weeks later, Mikey develops diarrhea, nauseous fever, and his mother takes him into the doctor. The pediatrician orders some routine tests and a fecal specimen. Here's the bacteria growing on McConkeys. The fact that it's growing means something. Where we have an arrow means that this is a colony of interest. And right there we have bacteria growing on McConkeys that's pink. And there's colonies that are totally white. And then these are mixed colonies. That's why they're both pink and white. So we have 2 cell types there. They're not isolated colonies. They then grow that up on an HE plate, and you can see the color here. That does mean something. Went too far, I think. Nope. And then there's the other results for case study 2. The results of the SIM tube, the results of the MR test, the results of the VP test, the results of the citrate test, and the results of the TSI test. And this one will be hard to discuss, but you can just say pink on the, so you can just say no. You can say K or minus on the slant, and then a plus or A on the butt. Hydrogen sulfide gas is being produced. I don't see cracking of the media, but let me blow that up really big to be certain of that. No cracking of the media means no gas being produced in fermentation. Yeah, no breaking of the media, no cracking. So no gas being produced, meaning hydrogen sulfide gas is not being made. Let me shrink that down. Go ahead and read about case study 3. Right there. Read about the tests that were run on case study 3. And there's some further tests and the results of these tests. Put those results of the tests in the table in the worksheet. Right here. Compare the results here to this table up there, and then identify the species, putting the species down here. And do that for each of the case studies. Any question about this lab? If there's no questions about this lab, go ahead and get started. And I will be here till about 8:45 to answer any questions. But once the last student logs out, I'm going to log out as well. Any questions about anything? If not, I'm going to pause the recording.